Got a pretty good crowd, so I'm going to go ahead and get started. Welcome everybody to the DCMB seminar series. Today we've got Dr. Magnus Rattray speaking with us from the University of Manchester. It's one good thing about COVID is since we're having these virtual seminars, we can bring people in from Europe very easily. So we're excited to have Dr. Rattray here. He graduated with his bachelor's in math and physics, and then ultimately his PhD in computer science from the University of Manchester. And then he did postdoctoral work on the statistical mechanics of neural networks at the Neural Computing Research Group at Aston University. And he was professor of machine learning and statistical bioinformatics at Sheffield University before returning to Manchester in 2012, where he currently is the director of the University of Manchester Data Science Institute. And Dr. Rattray uses probabilistic modeling and Bayesian inference techniques to study biological systems across a range of temporal and spatial scales, from gene expression in single cells to longitudinal population health data. And what's particularly impressive to me about his work is his deep knowledge of both statistical machine learning and biological applications. He's a world-class expert in Gaussian processes, and he's been working on genomic applications since the days of microarrays. And more recently, he's developing Gaussian process methods to uncover oscillations in single-cell imaging time course data and infer pseudo-time from single-cell RNA-seq data, which I believe he's going to talk about today. So Magnus, thanks very much for speaking with us, and we're excited for your talk.
Thanks, Joshua. Thanks for the nice bio, and you made me feel old now talking about microarrays. Okay, let me share my screen, and I hope that works. Is that okay?
Yep, looks good.
Brilliant. Okay, so thanks so much for inviting me. It's sad that I can't actually visit Ann Arbor. But I did have a look at it on Wikipedia so that I felt like I was actually in the place, you know. It's also unusual for me to give a talk just after putting the kids to bed. So that's also, but luckily they've gone okay. So hopefully they won't interrupt us. Okay, so I am going to talk about Gaussian processes. As Joshua says, I've been working on that for quite a long time. So just see if I can just move forward. So I'm going to give a quick sort of tutorial about Gaussian processes. So I think they're a useful statistical model that, you know, it's useful to know about in general. So hopefully that intro will give people a bit of a flavor about what they're about. And then I'm going to talk about 3 different ways that we've applied them recently. I'll start off talking about pseudo-temporal inference in the specific case where we have some capture time information from a single cell time series data. And the Bayesian nature of the Gaussian process method is useful to encode that prior information about capture times of the cells. And that's really building on other people's works, but our extension was to introduce some extra latent dimensions into that model to capture other variation, such as differentiation and branching. So then after talking about branching in high dimensional data, I'll go down to the gene level and talk about modeling branching at individual genes from single cell pseudo-temporal or pseudo-time course data. And finally, I'll just talk about a new extension where we've introduced a negative binomial likelihood into Gaussian process framework, which allows us to model count data a little bit better. And I'll talk about spatial inference with that type of model. So first, Gaussian processes 101. You know, if people want to ask a question, you can just unmute and go ahead, or put things in the chat, whatever. I don't mind. If there's something in the chat that I missed, maybe Josh can alert me. What's a Gaussian process? So it's a probability distribution, but it's a probability distribution over functions rather than numbers. So a Gaussian distribution is a distribution over numbers. A Gaussian process is a distribution over functions. And the functions encode our sort of prior beliefs, or they model structure in data. So for instance, they might model the fact that time series is very dynamic. Or they could model the fact that your time series could be stochastic, like from single cell live cell imaging traces, for instance. Or it could be smooth because you're averaging over millions of cells. It could be stationary, so it looks the same at different places, or it could be non-stationary. And the covariance function encodes the type of functions that we can draw from this probability distribution. So the covariance function really contains the sort of modeling strength of Gaussian processes. So why the Gaussian processes? What's the link to Gaussian distributions? Well, I'm going to try and explain that here. So the dots here are numbers from a 25-dimensional vector, which has been drawn from a multivariate normal. So you take your, multivariate normal random number generator from R or Python or whatever, and you draw a 25-dimensional vector, and then you plot the data, and the indices in the vector are along the x-axis, and the values in the vector are on the y-axis here. And I've chosen the covariance matrix of this normal distribution to have a specific structure where, so white is high here, and it means that indices that are close together are highly correlated. So this covariance matrix, or the variance is 1, so you can think of it as a correlation matrix. So things where the indices are close together, so if we take the 10th element and the 11th element in this vector, then if we go along and look at that box, it's high, and that means those two things are highly correlated. So if we draw that vector, it looks a little bit, join those dots, it's a smooth curve, okay? But it's not a function, it's a vector. But because we've chosen the covariance matrix in a particular way, it looks smooth. So Gaussian process is just where you take, instead of having n equals 25, you have n equals infinity. So and actually these curves here aren't really drawn with infinite number of points. I think I chose 400, so actually this is now a 400-dimensional vector, but it just looks like a function because everything is so smooth and close together. And now the covariance matrix, or the correlation matrix, is no longer a matrix, it's actually a function. So it's this thing here. And it's got the same form as this guy. It's high in the middle along this diagonal and falls off on the off diagonal. So that means this function here takes its maximum value when 2 points are right on top of each other, t equals t prime, and then it falls off otherwise. And if you draw functions from this particular covariance function, then they're going to look smooth. And there's a parameter of this, which is a length scale here. So it's t minus t prime over this length scale squared. If I make that large, then the functions cross the 0 line less often, and they have a longer length scale. If I make that small, then they're going to be more wiggly, and things are going to cross the 0 line a lot. So that parameter, we call it a hyperparameter. It tunes how wiggly these curves are. So Gaussian process is a probability distribution over wiggly curves. And the parameters of the covariance function, they tune how wiggly they are. And here at the beginning, there's an amplitude at outside this exponent, which says how much amplitude there is in these functions. So they're wiggly and they go up and down. Alpha tunes how high they go up and down, and L tunes their length scale. So that squared exponential covariance, that's one choice. This is what one sample from that looks like. Sometimes we call it an RBF, which is historically because it's related to radial basis functions. So that's one choice. If we change the function, instead of having a L2 norm here in exponent, but we have a L1, then we get stochastic SADs. And that's actually like Brown motion in a potential. So that's called an Ornstein-Uhlenbeck process. Also, sort of make more complex variance functions. So here I've one of these Ornstein-Uhlenbeck processes. I've multiplied it by a cosine, and that's induced these kind of stochastic pseudo-periodic kind of shapes here. So it's what you could call kind of, it's not completely periodic, but it's kind of stochastically periodic. And actually in the past, we've used that to model oscillations in single cell data, time course data, so longitudinal microscopy type data. I'm not going to talk about that today, but if you're interested, that's the paper. Okay, so Gaussian processes are distributions over functions. And the reason people in machine learning are interested in them is because they're very practical inference over data. So on the left here, I'm showing samples from our squared exponential distribution. So that means that these are smooth samples. And if we're doing Bayesian inference, we can think of these as our prior belief for what some time series is going to look like before we see data. So I haven't seen any data, but I know that, you know, it's a bulk RNA-seq time say. So I'm expecting things to be smooth because it's averaged over tissue. And I don't really know what's going to happen, but I've seen lots of time course in the past, and they always look like wiggly kind of curves like this. So that's my prior belief. And then I do 4 experiments. And have to be past closer data. In this case, I've done a super clean experiment, so these are really low noise. But in between the data, I don't really know what's happened. So the Gaussian process allows me to say, you know, the prior tells me what the functions look like, they're smooth. The data tells me, constrains me to have functions that pass closer data. And in between, the functions are allowed to vary, and that gives me my uncertainty in what the inferred functions should look like. So the kind of posterior distribution after seeing data is this kind of sample of functions that all pass close to the data. And that's that allows, and you can do all this analytically, it's fully tractable, and that's the nice thing about Gaussian processes. So you can do Bayesian inference for nonlinear regression exactly. And Josh was asking about Neil Lawrence, actually. So this is an example of the simplest thing you can do with Gaussian processes. You have some time series, gene expression data, and you just want to ask, is it changing in time? Or later on, I'm going to talk about space. Is it changing in some spatial dimensions? And the simplest thing you can do is just work out the probability under a dynamic model, so under Gaussian process. Take away the probability under a constant model, and that gives you a log likelihood ratio score for whether the gene is dynamic. And you can just use the data, find the dynamic genes, and then push them into the next sort of stage of data analysis. Now, Gaussian processes, if you just apply them naively, are slow because they involve a big matrix inversion, and that's cubic in complexity with the number of time points or the number of spatial locations. So that's completely impractical. But there's lots of ways of getting around that. So we use something called sparse inference with inducing points, and that scales linearly in the dimension of data and quadratically in the number of inducing points. But the inducing points only need to really be sort of logarithmic in the dimension. So k there can scale logarithmically usually, I think. And the whole thing is just a little bit slower than linear. So that's not bad. Sometimes we have a model that has some, I'm going to talk about models with binaries. I'm also talk about Non-Gaussian data. You can no longer do everything that case. And you have to use something like MCMC or variational inference. And we use variational inference. And finally, you can also benefit from all the advances in software engineering from the deep learning community. So they've got these really nice methods like TensorFlow and PyTorch. These methods allow you to make really good use of processors, so you can use all your CPUs and GPUs to full effect. And really, these things have matured tremendously in the last 5 to 10 years. So I think that Gaussian process inference is a lot more practical for big data than it was in the past. So I think it's really become a very practical tool. Okay, so that was kind of, you know, Gaussian processes 101, whirlwind tour. So now I'll talk about a few applications, bioinformatics applications. So first I'm going to talk a bit about dimensionality reduction and pseudotime inference. So people are probably very familiar with pseudotime. It's well established now, and there's tens of packages doing this. So the idea is, if we're doing single cell genomics, we typically destroy the cell when we measurement. We can't follow it in time. And if we're interested in something that's going on in the cell, we might want to infer where the cells are in that dynamic process. So it's a bit like rediscovering time from high dimensional time series data and having a sort of pseudo-temporal ordering of cells. And here's a picture from the nice slingshot method, where first you reduce the dimensionality of the data into some low dimensional space, not really 3, I think a bit higher than 3, but just this is just a picture. They do some clustering, and then they join the clusters together with a minimum spanning tree, and then do some smoothing, doing some sort of principal curves modeling through that space to work out pseudo-time profiles. So there was a nice paper from John Reed and Lawrence Vernish a few years ago introducing something called the DeLorean package, which did pseudotime using a Bayesian GPLVM, which is a type of Gaussian process. But it's a Gaussian process where you infer the time variable as well as the data y. So you're inferring y and t process at the same time. The nice thing about this is that if you have some prior knowledge about where the cells are in the dynamic process you're interested in, so maybe you've captured them at some particular point in the process, then you can introduce that your model as a prior, and that information about time helps you infer pseudotime. So you can't apply this to every data set, but if you have a data set where you have some capture time information, then this is a useful approach. But this was a nice paper, but it didn't really scale very well. And in 2016, the data sets weren't so huge. They used, I think, Stan to implement this, and every cell was a variable that you had to do MCMC over in Stan. So Stan's really nice, but it's not going to scale you up to millions of cells. So we re-implemented this using a more scalable architecture using GPFlow. And we introduced a new or a modified variational inference algorithm. And another thing we did was, as well as inferring pseudo-time, we also introduced other dimensions into the latent space. And we found that to be quite useful. So as well as having time, we also had some additional latent variable, call it X. And X could model other types of variation that might reflect, for instance, branching that's going on as you go through time. And we published this as the Grand Prix package. So just to explain the branching, I'm going to show results from, it's not a very big data set, but it's a very nice data set for sort of explaining the idea. So it's an old data set using qPCR on very early embryonic development in mouse. And the idea is that you go from really the earliest stage where you have a single cell, and then it goes into doubles into 2 cells, and 4 cells, 8 cells, and so on, into the inner cell mass, and then the trophectoderm, epiblast, and primitive endoderm. And so by the time you're at the 64 cell stage, you've got 3 different cell types. So if you can capture the cells at all these different stages, then you've got a lot of information about time, but then you can use pseudo-time to kind of model the fact that the cells are differentiating into different cell types over time. Now, if you just do PCA, it doesn't really work. It's clearly a nonlinear data set. And so PCA is not going to do it. Everything's going to be kind of mixed up. The standard GPLVM is a typical kind of nonlinear dimensionality reduction algorithm. And so you can think of that as just doing tsne or UMAP or one of these kind of methods. And it's nonlinear, so it's much better at separating out the different stages. So here I've used colors for each different stage, and then colors also for different cell types. But what you can see is that the time order of the process, so these differentiation times, they aren't really reflected in the latent space. So this low dimensional space here doesn't really capture the fact that you go from 8, 16 to 32 to 64. The 32 inner cell mass is away from the trophectoderm at 32, and it's separated by these other cells at 64 stage. So the latent space doesn't really reflect the temporal nature of the data. So on the left, I've just shown the same standard GPLVM picture. On the right, I've shown the result of using Grand Prix. So with Grand Prix, we put some prior knowledge about the fact that we know which cells are at. So we know the 1, 2, 4, 8, 16, 32, 64. And then we just use GPLVM to add in the additional variation in the, here's the y-axis. Really captures the differentiation into cell types at time 2, time 64. So it gives us a much kind of clearer representation of this developmental process over time than the standard representation. Another advantage of adding in that extra dimension, so you can go back and look at how good your latent representation of the data really corresponds to the known times for the cells, so the capture stages. And if you use the original approach of just inferring time, only having time as your latent dimension and no other variation, you actually get a much poorer solution. You get stuck in local optima basically, which don't really reflect the temporal progression through those stages. But if you do the 2D optimization where you also allow for the branching, then the times are much better reflected in the latent space. So it seems like having this 2D approach rather than just learning a 1D space also improves the 1D inference, improves the time inference. And then once you've inferred pseudo-time, you can go back to individual genes and see how they behave. So, you know, in this case, we can see that ID2 here is already looking like it's differentiating at the 16 cell stage, even though that wasn't apparent in the kind of global reduced dimension picture. But it's probably not involved here in the epiblast differentiation later, whereas SOX2 is involved in the epiblast later. So now that kind of links me to the next part, because if we look at individual genes, as we are here in the bottom, then we might ask the question, which genes are associated with branching events? And so we can see here that SOX2 is clearly involved in that transition from the inner cell mass to the epiblast. And so in order to do that, we have to go down and kind of model things at individual genes. And that's what I'm going to talk about next. Just before I finish, scalability. So that was a very small example. As I said, introducing this implementation in GPFlow allows us to really scale this up to big data sets. This is not a pseudo-time one. This is just a dimensionality reduction problem. So this is a standard GPLVM, but it shows that it's very kind of scalable. And this data, I think we initialized using TSNE, and what we found was that the GPLVM latent space actually ended up more consistent with the clusters in this data than the TSNE for this one, where the clusters were found not in the low dimensional space, but in the higher dimensional space. One nice thing we found was the, so Uber developed this package called Pyro for probabilistic programming. It's a bit like Stan, but it uses different inference techniques. So you can just write a model and automates all the inference part. It's really nice. And we just used our paper to make the example for the GPLVM. So that's completely independent. We just noticed they've done this. And they've implemented it in both from scratch. And they get exactly the same results. So that's quite nice because we're very much into repro research and reproducing results. But reproducing something, including implementing it in a different framework and getting the same results is very nice. So that gave us so it shows actually the model's quite simple and can be implemented in many different ways. And, there's lots of practical tools out there for doing this type of modeling. And Pyro looks actually really nice. Recently, Verma and Barbara Engelhardt have a really nice version of the GPLVM, which is a bit more robust than the standard Gaussian that we use. So they're using a student T likelihood. So it's a bit more robust to heavy-tailed kind of noise. They use a slightly more general set of covariance functions. And it looks really nice. And if you look at this paper, it actually shows that this scales better than things like UMAP and TSNE actually. So I think people are probably thinking Gaussian processes are a slow thing and maybe avoid them for that reason. But if you look at this paper, you can see actually that when you implement them in a modern framework, they're pretty scalable. So I like this paper. I thought it was really nice. Okay, so that was kind of looking at GPs for modeling high dimensional data and inferring a low dimensional latent space. I'm going to go back to low-dimensional data, modeling actual individual genes, and looking a bit more at that branching question, seeing if there's evidence for genes branching and whether they branch early or not. So originally we had a model for branching gain processes, which we used for time series data. And it was for what's called a two-sample inference problem, where you have two different time series data, and you want to understand the differences between those two time series. So for example, you might have a time series for a wild type and a time series for a mutant organism, and you follow the genes over time and you ask, which genes are changing in the mutant and when are the changes happening? And that's the question we addressed originally with this work from a few years ago. But we can also apply this to single cell pseudo-temporal data. So a Gaussian process, as I said, is a distribution over functions. But now I'm going to talk about branching functions. So here, the red function is a standard sample from a smooth Gaussian process. And the blue one is a Gaussian process that touches the red one at a particular time point and then goes off and does its own thing. So here's one sample from that distribution. Here's another. Here's another. These are just all samples from the same covariance function. And, so that's the samples from that distribution. And then you want to do some inference over that. So you plug that covariance function into your Gaussian process inference tool, and you can make inferences about where, when you have real data, where does the branching happen? So two things you can do. You can say, what's the probability of branching at each time in that time course? And the second thing you get is, what's the probability of branching at all? What's the probability of branching versus non-branching? And we can work out that using a Bayes factor. So if I look at time series, so this is some time series data. Then at the top, I'm showing the probability of where the branching is. So the posterior probability of where those two things are beginning to diverge. There's no evidence. So there's very low evidence for those two things not branching. Here's an example where there's strong evidence for these things not branching. So they look identical over time statistically. At the top is the posterior probability of where the branching is, but the highest value is at the end. And the Bayes factor for whether it's branching or not branching is basically, it's worked out as the height of the last bar divided by the average height of all the bars. So if the last bar is higher than all the other ones, it means that the most likely thing is that there's no branching. So that was for time series. That's kind of old work. Then when we have pseudo-temporal data, we have some additional things. So first we have to work out time. I talked about that before. Then we have to work out, which branch does the cell actually belong to. We have to work out which genes are involved in the branching and when they change. So the last two are the same as for time series, but the question of where the cell lies on the branch is actually a new problem, and we address that in this BP package. So a method that did something like this was called BEAM, which is based on splines. And BEAM was, it was implemented in earlier versions of Monocle, but I don't think it's in the current version. So if the data is low noise, and you can see that so BEAM here is on the left. We have we've done pseudo-time inference. There's some global branching, which is the dashed, the black line here. And then the actual branching for this gene is a bit later than the global branching, and we want to infer where that is. Beam fits these splines, it looks at where they cross, and it finds that they branch late. This is synthetic data, so we know that this is close to the truth. BGP does something similar, works out that the branching is here, has some uncertainty, but generally the two methods agree. However, if there's a lot of noise, which is more typical with single cell data, then the Gaussian process says, well, you know, the most likely branching is still late, but I'm super unconfident about it. So I'm going to put big error bars or credible regions. Whereas the spline doesn't have any concept of uncertainty, and it tends to be highly biased towards this global branching, this black line. So it becomes very conservative and says, I think this gene just branches where the global branching of all the other genes is. The other thing that the Gaussian process allows us to do is it allows us to deal with the situation where branching is actually earlier than the global branching in the data. So if we use some sort of method like slingshot or monocle, and we work out where the global branching of the cells is, if we have a gene that branches very early, then often it will branch before that global branching in pseudotime. And we can see here that the purple dots aren't labeled to which branch they belong to. And the GP allows us to make inferences about which of these branches the purple dots belong to. Whereas the kind of beam approach doesn't allow us to do that. It actually just randomly assigns the purple ones to either branch and fits the spline. So in that case, so the red pluses here are beam, they're kind of biased towards this global branching in this example. Whereas the blue and black ones are, the black is the ground truth and the blue is the BGP result. The blue and black don't match here because pseudo-time can be a kind of nonlinear function of actual time, and there's always a risk that you're going to be a bit biased. So I think what's happened here is pseudo-time is a bit warped relative to actual time, but the kind of rank order of the cells in pseudo-time is probably okay. So looking at some real data, from bone marrow myeloid progenitor populations, so the way you do this, you apply some general approach for pseudotime and branching inference for the cells. And then you can look at individual cells and you can score them in terms of their evidence of branching at a particular branching in the sort of global tree. And then you can look at the locations. So here's some examples in real data. These are kind of genes with strong evidence of branching in that data. And BGP, the posterior for where the branching is at the bottom here. So most of these are early branches, but we can see that CAR2 and CAR1 are kind of branching a bit later here. And then we can get a confident gene ordering. So where things here are connected by line, that means that they're confidently branching later or earlier than the gene they're connected to. So it allows you to kind of rank order the genes. Okay, so finally, I'm just going to talk a little bit about some recent work where we've extended these types of Gaussian process models to modeling counts data. So here's an example where Gaussian processes have gone wrong a little bit using a standard Gaussian likelihood. So in the bottom here, I've done the same thing of transforming log counts plus a constant and modeling that using GP. And then I'm trying to do a two-sample test to work out whether these two time series are different. And they are different. I mean, or there's strong evidence in the top here when we use a negative binomial likelihood, a proper kind of counts likelihood. But in the bottom here, we find that the Gaussian is not fitting one of the time series very well. And it's probably confused by the fact that we don't have a lot of replicates here, and the replicates just happen to be very close together in this example. And so the GP is somewhat confused, and it's kind of wiggling through this data and just not fitting well. So if we're doing a two-sample test, this is actually this model, because the green fit for the bottom right there is not good. It means the model is going to prefer to say that there's no difference between green and blue in that case. So the inference has kind of failed, I think. Whereas in the top, the negative binomial fit to these time series really captures the fact that these are two different functions. They're both smooth. They both look like they've got reasonable, credible regions. And it's more plausible to say that those two time series are different. So sometimes the Gaussian likelihood goes wrong because it doesn't correctly model the noise distribution in the data. Now, there's been quite a few papers suggesting that negative binomial is quite a good choice for single-cell RNA-seq data. And I'm not going to kind of go into the details of those discussions. We've actually implemented negative binomial and zero-inflated, but in practice, we find the zero-inflated doesn't add much in most applications. So the idea of a negative binomial is that it models counts data which has excessive variance or dispersion relative to a Poisson distribution. So the variance in the data is equal to the mean plus an extra term which captures the dispersion. And in the Gaussian process, what we do is we model the log of the mean of the data as a function from a Gaussian process. So the dispersion models the counts data, and the mean of that is modeled using the Gaussian process. And if we use synthetic data, we can look at, is that useful? Well, if you have low dispersion, then the Gaussian is pretty good, So the Gaussian and the negative binomial are quite comparable for low dispersion data. The Poisson works reasonably well, but for high counts, it can still break down a bit, even with quite low dispersion. But when you have high dispersion, especially if you have high counts and high dispersion, you can see quite a big difference between the negative binomial and the Gaussian. And here what we're doing is one of those one sample tests where we're just testing to see if there's dynamics in the data. That's kind of the type of statistical test that we're doing in this comparison. So it seems like negative binomial is useful in the case where we have high dispersion data. And if we have this type of single cell pseudo-time series, for instance, we have high dispersion, but also if we have spatial transcriptomics data, we also see high dispersion often. Apply this to spatial data just to a very simple problem of identifying differentially expressed genes spatially. Spatial D package does that with the standard Gaussian process, with the Gaussian likelihood function. And we've just plugged in a negative binomial likelihood into the same pipeline. So everything else is very similar. So the idea here is that if you have spatial variation, then those should have a characteristic length scale. So you should be able to model them with a covariance function that has a kind of length scale parameter. If you have non-spatial variation, it should be better modeled just as a noise, and it won't be captured by a kind of spatial covariance function. So we're doing a likelihood ratio test between a model which is flat and only has non-spatial variation versus a model that has spatial variation to see which is best. And what we find is that having this negative binomial likelihood function really gives us much better sensitivity and also avoids some rather implausible positives. So Spatial DE in this Venn diagram here is identifying a smaller number of genes as differentially expressed in this data set. And then the ones which spatial DE says are positive, when we look at them, often they have extremely small numbers of counts. So it looks like the fact that if you have very small numbers of counts and use a Gaussian noise model, then you don't have a lot of information to estimate things like variance when you have that little data. So it's rather implausible to use a Gaussian noise model in that case. The counts likelihood is quite nice, because in the case where you have very little data, you're just going to basically infer something like a Poisson or slightly over dispersed Poisson. And then you get a lot of information about variation in the data that you wouldn't get from a Gaussian model. And if you look at some of the ones which are positive, you can see that they kind of are reflected in images which are independent from the spatial transcriptomics data. We've also scaled this up using the sparse inference techniques I was talking about, and I was hoping to present some results on SlideSeq. We've run it on that data, but we haven't got results that are ready to present. But with the sparse methods, it scales up to large scale things like SlideSeq and 10x spatial data. So I'm done. So just to summarize, GPs they're very natural for modeling branching in time series. Uncertainty allows you to kind of get a confidence in ordering your events. So if you want to know which genes are branching early or late, you can order those with a level of uncertainty. To be practical for single cell, you really have to use speed up. So we use these kind of variational inference techniques in the GP flow package, which is very good for that. And recently we've introduced negative binomial likelihood into these Gaussian process methods, which I think gives us a much more realistic model for counts data. And we've been applying that specifically in the spatial inference. Challenges, well, I separated out pseudo-time and branching. It would be nicer to model them together and have a kind of joint model which also has pseudo-time and branching at the same time. And we'd like to look at more complex spatial models. So we're interested in point processes, and also when you're fitting these models to, and sometimes we're fitting them to all the genes. So we're fitting 10,000 models. It's really important that the inference is robust and that hyperparameter estimation is robust. So you have to catch numerical issues. You have to restart if you converge at a bad local optima. So those type of things we're working on, but I think we can always improve so that you don't get kind of bad inference in some bad cases. And finally, I'd like to thank people who did a lot of the work. So Suman Ahmed and Alexis Bukavalis did a lot of work on Grand Prix and the BGP method. Jing Yang also on the branching. Esti John and Socratia worked on the spatial and the negative binomial, and Nuha as well, who's put the GP counts package together. And then James Hensman is a longtime collaborator who I've worked with on this work. Thank you.
Great, beautiful work, Magnus. Thank you. All right, so now we have time for questions. So if you have a question, feel free to type it in the chat or just unmute yourself and ask. And maybe I'll ask a question while people are thinking. So these Gaussian process models are beautiful and elegant. I wonder, so the other sort of technique that people are using now for a lot of single cell problems is deep learning in various forms. Like, so I wonder, what do you think are the pros and cons of GP regression versus ML, multi-layer perceptron, or GPLVM versus variational autoencoder for some of these applications?
It's a really good question. My group also uses deep learning and variational autoencoders, and they're very nice. I think that let me take the variational autoencoder versus GPLVMs one. One advantage, I think, of the GPLVM is that it has a very explicit model which allows you to introduce prior information if you have it about the latent space. So I gave a simple example of that, but I think you can probably think of more complex models where you have some prior information that you want to build into the latent space. And I think the GPLVM provides a really nice way of doing that. I think in terms of deep learning is extremely powerful in terms of nonlinear function estimation, basically, and has great power. But I think that it's quite hard to build the uncertainty into deep learning well, and calibrating the uncertainties in deep learning models is quite tricky. I mean, people work on it, so I'm not saying it's not something that you can do. It's an interesting area. But I think the benefits of using GPs is a very explicit treatment of uncertainty and a very explicit treatment of priors and prior knowledge. But, you've got to use the right approach for the right problem, and we use deep learning all the time. So it's also a great set of tools.
Looks like we've got a question in the chat. Chenli asks, is GP flow easily transformed and applied to other single cell modalities to find branching and pseudo-time ordering?
So like an example, single cell ataxic or something like that, perhaps, or other, is that the other types of omics and so on?
Perhaps? Yeah, I think that's how I would interpret that question.
That's a good question. We actually work a lot on single cell ataxic data, often with ataxic data. So one of the challenges there is because the data it's not quite binary, but it's almost binary. It's got a little bit less dynamic range than single cell RNA-seq data. So it's a little bit more challenging to normalize out library size effects in single cell ataxic data, we find. And having a probabilistic model for modeling the library depth is really nice. So you can model the kind of observation process, like how many reads you get from the experiment, on top of the well, you might model the data as binary, or you might model it with some other model that's not binary. We often just use something binary. And you can put that type of likelihood model into a GP. So I think that's one of the nice things about GPs, because they have a very explicit probabilistic model. If you want to plug in some other probabilistic model that models something like the library depth, you can do that actually. And I bet the Pyro package would be quite good for that, because you could just write things down like you do in Stan, and the inference would the machine just turns the handle. So yeah, I think definitely.
Any other questions? One other thing I was thinking about as sort of a tool in your set of Gaussian process tools is multi-output GPs. Have you guys investigated using those for anything? Maybe like covariance relationships among genes?
Yeah, that's a really interesting question. So exactly. We're very interested in that in the spatial case, because I think discovering latent lower dimensional GPs and relating them, yeah, that's a very good question. I didn't talk about that type of thing, but that's exactly one of the extensions. I mentioned point processes, but also multi-output. Actually, in the past, in older work on time series, we use GP model different layers of transcription. You know, modeling pre-A's, A's, and proteins, and degradation and production models. And you can also do that with multi-output GPs. So yeah, I think multi-output GPs have great potential.
Great. Well, it doesn't look like there are any other questions, so I'll let everybody go. Thanks again.